The title is the Emirati Project, where long reads became an integral part of large genome projects at first. And I work at G42 Healthcare at UAE. And G42 Healthcare recently merged with Mubadala. So Mubadala Health is now one of the first integrated genomes healthcare in the region, and it has why it is very relevant in the context of the Genome Project is that Mubadala Health is M42 is now the integrated healthcare system. It has hospitals, clinical diagnostics, IVFs, and it has a genome program. It has a CRO, and so it's the idea that genomics needs to be within the healthcare system. And that's actually what I think we all thrive for, is to go for things that are very cool, like all the posters out there, but to actually make an impact. And this is something that we have been trying to do as a community for many years, and I think we are reaching that point where we are going to really see the impact, but being integrated into the healthcare like M42 is, would be very significant. And so that organization has several hospitals, has 21,000 people there, and so on. Now, this is Abu Dhabi, and this is Abu Dhabi, as you imagine, 50 years ago. This is the main civic center of Abu Dhabi. This is Abu Dhabi now, taking from there, and you can see a big difference, right? This is 50 years, 50 years. So you went from this desert to there, with this wonderful sophistication that you can see in people in Abu Dhabi. And that is because, of course, there is this great oil, but also because of this man, the founding father of the UAE, which is actually one of the major political figures 20th century for autodetermination. I think he's actually, I have to admit, before coming to UAE, I didn't know that much about him, but it's really worth investigating because he's an amazing man that wanted to get science and knowledge integrated really into the diversification of the economy. So this man said many things and always about knowledge, science, how you get that for the good of the people. It's actually extraordinary. And the current leaders from Abu Dhabi and UAE, two of the 7 Emirates, President and Vice President UAE, are exactly aligned to that. Science, knowledge, and the good of the people. So they were convinced, and they pushed forward for a very large Emirati genome with the name Future of Those Generations that wants to sequence all the 100 million Emirati in the population, which is quite extraordinary. I remember being in college when sequencing was something, and now we are sequencing 1 million people, which is extraordinary. So why? I mean, first line, for a better care, for better health care, which again goes back to what we want to do, which is really put it up there. We'll really put it in the sequencing and the genomics information into clinical. Also, because we is the 6th country with the highest birth defects in the world, so that's kind of a plot, and UAE is up there on the 6th on the red line, and the UK and France, for instance, are very much at the bottom of that. And so that, of course, brings a high burden in suffering and also in healthcare costs, and that's where this program wants to be very much, is to reduce healthcare and to reduce human suffering by reducing disease, right? So reduce healthcare, high costs, genetic insights leading to treatments, cures for diseases, and of course bringing business opportunities to UAE. And this is actually very much also ingrained into what this program is to make sure that innovation, business opportunities, piggybacks on this massive effort of sequencing. So why is it unique, this program? So first, because there is also very, very deep phenotype data coming from an integrated system of electronic health records, right? So that means that there is deep phenotype from the genomics linking with the phenotype, which is what we need because genomics on its own doesn't mean anything. Of course, we all know that we need phenotype. Then, from the beginning, strong link to disease ab initio with the regulators and consent, and this is unlike other programs, right? I've seen Gary Stephenson from Decode mention his frustration that although he knows that many people, many women namely, would have breast cancer, he is unable to tell that information to those women at risk. And this program, in that sense, DGP is very different because it is built in so that people can use that information. Then, obviously, it's the first that I've studied at this scale, one of the largest non-European, which is very significant to expand genetic diversity. And then two specific details from this population that make it very interesting from the power of genetics. One is that there was a massive expansion on just 2 or 3 generations from 45,000 people maybe to 1 million currently, which means that there is a lot of founder effects. If someone on those 44,000 people had one genome, of course, that's going to translate into many people now, bringing more power because instead of just one person, the signal would be just bigger because there will be more people there, right? And then, by the tribal nature of the Emirati population, high levels of consanguinity, also increasing the signal that will be able to genetically to find this, and also to bring a mix of population genetics with family structure, which means that the algorithms, even in terms of mixed models and so on, could be very interesting, and also increase the power to detect pathogenic variants. And then, 3 sequence platforms from the beginning: long read, short read, and MGI and Illumina, and long read for NT, and then the largest read in methylation, of course, piggybacking on the fact that we have the nanopore. Now, Tony, you told me not to put a lot of text in the slides, so as you can see, that's like how well I followed that instructions. But this is actually very important, right? Because this is the impact of DGP. And when you think about the massive amount of genome sequencing, 1 million people, plus the money, plus the infrastructure, plus of that, of course, there is not a single stakeholders. We are talking about several stakeholders, and we are talking about many outcomes that we are going to get out of this. Right, volunteers, the first, of course, short reports, specific genes. Yeah, there is a few reports directly to the people, and application as well, pharmacogenomics on relevant genes. And then this may be the bigger in terms of the healthcare system. Population genomics goes clinical. The idea here is population genomics goes clinical. It's just not population genomics for its sake. And so several projects: pre-marital screen, pre-IVF, newborn genetic screening, pharmacogenomics used in the clinic, rare disease causal genes, genetic screenings for rare variants, and fast-track genetic testing. See, these are all programs that are already in place. Of course, by the nature of what we are trying to do, there is much more that is going on. It's not yet fully developed. It's an ongoing process, but these are already projects that are going on. I'd like just to few words on this premarital screen. Actually, Brad from Oxford mentioned a bit that type of work, which is very impressive, by the way. So what we are doing now is the UAE has a program for counseling as well, for premarital, for couples that want to get married, they have kids, of course. And so what we do now on top of DGP is to get the two genomes, understand whether there is any recessive mutations coming on. There is a few recessive mutations that are more relevant in UAE. And then the couples are counseled. This is Sari Islam, one of the counselors that are integrated into biogenics. And then she counsels the couples, right? And so the couples would have all the options. One of them is to pre-IVF, which is also included in our services. And that is how the idea of the human suffering that I was talking about is the idea that we could move forward from there. So, research. One big project is Emirati reference genome. That project is led by Dr. Abiba Al-Safar from Khalif University, which is doing reference genome for the Emirati population. Then HLA calling, collaboration with Fritz Sedlacek, which I'm going to mention just in a few slides. And then other projects, namely Sarah Sullivan on genetics associated with successful outcomes of bariatric surgery. And all of this means, of course, that we are open for that because we are very interested in getting those collaborations on top of this data set. And then G42 is a very strong AI company driven that was in the genesis of the company. And so a big project as well is doing AI research on outcomes based on phenotype genotypic inputs. ChatGPT, of course, generational models are all over the place these days. And so it is the idea that with this information and others, we'll be able to generate some hypotheses based on when the patient comes to the clinic, what would be the suggestions from the clinician to move. That's a project that we are right now starting. Of course, pharma biotech would be interested in this data set. We imagine it expands diversity, input diversity in the global stage, something that even the FDA now is forcing on all clinical trials. And so we believe that this data set will be very relevant in that context as well. And of course, pre-discovery at population level with GWAS, FEWAS. Again, the idea that because this is a family structure that is imposed on this data, that we could have more powerful algorithms around this. Now, why does G42 believe in long reads? And why, in December 2019, was it signed a big deal, and Gordon was in Abu Dhabi, Ashish Koshi, the CEO of G42, which is somewhere, which I saw him already before, so he must be in the audience, signed this deal. So in December 2019, the start of VGP started with long reads. This is not an afterthought, this is not, we started directly doing long reads, and of course for this audience, that's very easy to understand, about structural variants, about phasing, about methylation, and also I think something that links very much with the idea of the UAE and the leadership of the UAE about the future, the idea that this is the future, that this is going to be the best, that this is going to be the encompassing. And so that's, I think, is also very much ingrained in the idea that we want to do long reads from the beginning. So 84,000 samples so far, sequenced so far, which is the longest as far as we know, and I think it is the longest from what NT tells us. 84,000 samples sequenced in April 2023. And by the way, this is implementing while COVID was going on. So the lab, the building of the lab, the machines, the GPUs, the linking with the setting up the server and so on was doing during COVID on these 2.5 years, which is, I think, pretty impressive. So anyway, 41 Prometheans is what we have in the labs, 41 Y100s now, 47,000 samples a year comfortably. G42 Cloud is what uses currently for the processing, although much less when the Y100s now are linked up to the sequencers, and 17 scientists in the lab and 3 bioinformaticians for infra support. Now, we have learned a lot, right? To do this many genomes, we learned a lot. This is a bunch of the things that we have learned. I'm not going to tell that much just because my ticking time is telling me that I should move forward. A couple of things, of course, we need to move from Guppy4 to Guppy6, now to Dorado. We implemented we implement SOUP as a model in Core3. We use Minimap from Sentient to accelerate because it's much faster, and many other little details there that we can discuss if you want. Now, a plug to my commercial team. I hope to gain, they are listening, and I'll get brownie points from when I need them. Of course, that we do this many genomes, not only for AGP. We are a commercial sequencing company. We are also ready to apply this to other population, large implementation at scale. So talk to us because we are open for business in that way. And I gained a lot of brownie points now, which I'm very happy with. This is Fatma Al-Kawouri, which is the director of the unit from ONT. And she's always smiling, as I can see, which is great. So now a bit more on the technical side. This was a joint work led by Stefan Ditford from AWS, Toruk Otzurk, Guilherme Copini from G42, Javier from G42, and Chris Simmer from Nanopore. And this is published 1 hour ago. I didn't try to get, now that I didn't even get my last presentation, but I wanted to put the link, the URL link, because it was 1 hour ago this was published, now on the AWS site as a technical post. So this is to show that Dorado achieves at least 2 times the speed of a base calling. Actually, this is many configurations of AWS. On the bigger machine, actually, it's 8 or 9 times faster, which is actually very impressive. This is this panel here. This is on methylation, because without methylation, it's not that much advantages, of course, because the Dorado is precisely bringing a lot of things with forwarding in CPUs now than GPUs. So this is where you get the most improvement, and which is great. Also, interestingly, for 5HMC, you can now call. Before, we didn't even bother benchmarking. It was so slow that it was just impossible for us to do it. But right now, with some configurations, we can indeed do, which is great, because that opens up other epigenetics configurations that before were just not even available because it was so costly to do. Now, what I'm going to show is just on 1,000 genomes. This is work that is going on, of course. We are now expanding from 1,000 to 10,000. We are doing the structural variants of the population. We are linking it to where the common is, linking with the family work, understanding what are the genes that are within those variants, linking it to phenotype. That's the current work. What I'm going to show is just on 1,000 genomes, a very limited part of this. This is, again, work with Fritz. This is Sniffles 1 with Sniffles 2. See Sniffles 2 actually, no, sorry. This is joint calling versus not joint calling. And what we can see is that joint calling in Sniffles 2 actually recovers quite a few variants, which is great to see. And, but it's slow. And so to do this with the 10,000, we need much more. And this is what Fritz is working on. I think even hire the software developer to do this for 10,000 genomes. And this is type of new algorithms that you need when you are indeed trying to do this at this massive scale. And I'm going to move quickly, sorry, to the next slide. This is the first structural variation characterization of the Emirati population, which is always a beautiful plot, because here we are seeing for the old chromosomes, for a full population, what's happening in the genome. We see this is just large deletions over 10K. We see some hits that indicate that there is some common population, some common variants. Now we are then in process understanding what are the genes that are underlying that, the link to the population, to the structure, and hopefully next year I'll have much more detailed information on this. And this is the bioinformatics team that is involved in the mapping, in the production, and in the several analysis needed here. So, on the same 1,000 genomes, for the HLA, the idea here is to do, again, a first characterization of population. What we find is that for HLA genes in class 1, A, B, and C are very polymorphic, and they are diverse. So none of them, this will be these ones, none of them are reaching very high frequencies, so they are not fixed in population. On the contrary, for G and F, you see very high frequencies, and one of them basically fixed in population. Of course, it's just 1,000 genomes, but we are seeing all these patterns that we are now going to investigate much more. And this is, we are seeing the same on the quality population that was published for 400 genomes. And so it seems there is good agreement, much more to come. Again, this is preliminary work. Here, it's something that someone is mentioning, how Nanopore is flexible, how it can be integrated into some workflow. For instance, you can imagine on some GP that would have this machine. And so this is seeing whether buccal swab and saliva would make a good correlation with blood. It does. We are now interested also to go lower, and this is, Fritz has this idea that you want to see how the coverage goes low. The work was shown before with PRS is at 1x coverage, kind of goes into that idea. Kind of, can we do this quick? Can we do this cheap? Can we do this fast? Can we implement this in the clinic? And that's what we are trying to do here. So, thank you for the people that did this, right? And the people that did this are basically the volunteers, right? So when I was preparing for this talk, I went to the outreach team, which is the most enthusiastic for G42. They're actually amazing. They really feel this as a mission, and they are really out there to get people to teach about genomics, how this is going to help the country and so on. And amid the tea and Arabic sweets and whatever, which was a very cool experience, they told me that once they got a call from someone at Al Ain, which is the Oasis of Abu Dhabi, and they went it was Friday, so the family was united. I think they are united every Friday for the prayer day, and they went there to collect because this family really wanted to be part of the Genome Project, right? And they collected 108 samples. So they went there, and then they collect 108 samples from a single outing to get samples, right? And I think this is the type of enthusiasm that this program has been doing in the Emirati people, and that's why I think that this would be successful and also an example of whether genomics is really going to go clinical and is really going to make an impact in the people's lives, which is what we all want in this room, surely for many years now. So that's it. Thanks so much for the presentation.